Hi there, and welcome to Chapter 3, Molecules of Life. I'm going to start out by showing you a couple of documents that you can use to follow along with the lecture to organize your notes. One of them is more of a note sheet, and the other contains some application questions that actually have come from your lecture textbook. So here we go. I'd like you to start by organizing a sheet of note paper or maybe a Word document with a 4x4 table, and you're going to call it Molecules of Life Macromolecule Table. You're going to put the following macromolecules each in their own row: proteins, nucleic acids, carbohydrates, lipids. And then in the 4 drop-down columns, you're going to name each of those headings monomer, polymer, where found or examples, and then any additional notes that you want to keep track of. So after today's lecture, you should have a better understanding of how the 4 big molecules are formed. Also proteins, and how nucleic acids are formed, and carbohydrates, as well as lipids. If you remember from the Chapter 3 lecture, all things are made of atoms and molecules. So in terms of biology, organic molecules make up us. We are actually mostly carbon, and you've heard there's a lot of carbon in the atmosphere, excess carbon even. So there's a connection for us to start out with. At the core of our carbon base, we actually have functional groups that are in the form of other atoms. So we're going to talk about a few main elements in this lecture, all related to the main molecules that make us up. Each molecule is kind of like a puzzle, and puzzles are made of many smaller pieces, just as the 4 macromolecules are that you can find in us. So the 4 main ones are proteins, like protein that makes up our hair and our nails, and even proteins that act as enzymes in chemical reactions. There's also nucleic acids, things like DNA and RNA. And carbohydrates, very common ones that we probably eat a lot of, is our glucose and sucrose and fructose and lactose. And then the 4th type of macromolecule is kind of a macromolecule, and we're going to count it here, lipids, so things like fats and oils. And so these macromolecules are actually made of smaller units, so those individual little puzzle pieces are what we would call monomers, whereas the whole puzzle, or the macromolecule, is called a polymer. All macromolecules are actually formed the same way, and that's through a process we call dehydration synthesis. So to dehydrate means to remove water. So if you see right here, water has actually been removed between this covalent bond in this polymer right here. So water was removed, and the rest of this polymer was spliced together. So synthesis means to combine or to make. The opposite is when molecules need to be broken down. And this happens, for example, when we digest the food that we eat. So we digest, pieces of carbohydrates, and those carbohydrates get broken down into their monomers, or those puzzle pieces. And how that happens is water actually comes in to this polymer and breaks apart this covalent bond right here, and water inserts itself between these 2 monomers. So that this is something that we call hydrolysis. And so hydro, think water, and lysis, you can think kind of breaking open, like to lyse, to break, and inserting water. So this is how we are going to break down the molecules that we eat in order to really get the nutrients and, in fact, the matter from them. The first macromolecule we'll go over is proteins. And so if protein is the puzzle, then the puzzle pieces are subunits that we call amino acids. There are 20 amino acids in existence. Our body makes approximately half of those amino acids. So the other remaining ones are ones that we need to get in the food that we eat. So if our bodies or our cells need to make more proteins and need certain amino acids, Some of them can be synthesized by us, but others we have to take in from the food that we eat. The basic structure of an amino acid is an amine or amino group on one end, and you see that carbon and that hydrogen right in the middle there, because remember we're mostly carbon, and then you see a carboxyl, so carbon and oxygen and a hydroxide also. So that's the gist of an amino acid chemically. When two or more amino acids are held together by a peptide bond, there you have protein. That was a goofy joke, I know. But an assembled protein or polymer is something we call a polypeptide. So think poly means many, and peptide is the type of bond that holds those amino acids together, forming a protein. Here's a great breakdown of how polymer proteins are actually assembled. So it's a dehydration synthesis like we saw earlier. So a hydroxide group and a hydrogen ion are removed in the upper left between those two amino acids. Water is removed, so dehydrate, and then synthesis means put together. So after we dehydrate the ends of each of these amino acids, we're left with this growing polypeptide or protein chain. In science, we talk a lot about structure and function, which is the form of something, and then what that form allows it to do. So in proteins, there are some general levels of protein structure, and we're going to talk about several of them here. The primary structure of proteins is super important, and what it comes down to is the order of the amino acids that make up that polypeptide. So this order determines all other levels of the protein structure. And what I mean by order is whether or not the amino acids in that protein are hydrophilic or hydrophobic. So if each amino acid is either of the two. So if you have a hydrophobic amino acid in your polypeptide chain, that amino acid is going to face inward on that protein. Hydrophilic ones are going to face outward. And hang on here for a second until I show you what I mean by this inward-outward. So the initial folding that proteins take on is something that we call secondary structure. So proteins can either be associated on the left there in beta-pleated sheets, or on the right as alpha helices based on which amino acids they have and where they're placed in that order in the chain. So the last level that we're going to talk about, the tertiary structure of proteins, is in this picture right down here. The shape of this protein is what it is because if you go into that zoomed in little box right there, down here, you can see hydrophilic amino acids are going to face outward on this molecule. Hydrophobic ones are going to face inward because surrounding this protein is water. Most of a cell is water and most of us is water. So remember that hydrophobic, hydrophilic relationship that water has and that other things have with water. Something that can really mess up the shape and the function of a protein is something we call denaturing, and how that happens could be the result of a temperature change, a pH change, and hydrogen bonds between the amino acids giving the protein that shape can get disrupted. And so when a protein is denatured, it becomes inactive, it basically comes apart. And so that's not what we want if we're actually in the market of looking for functional proteins for our cells. Back to the idea of form and function, proteins can only function based on what their shape is. So, there are protein markers on cells that allow cells to identify other cells as good or as intruders. And for example, there's proteins on red blood cells that determine which type of blood that you have. So those proteins allow your blood to know if the blood coming into you in a transfusion is acceptable or if it's going to cause you to clot and clump and possibly be really detrimental for you. So it all comes down to shape and job, or form and function. The next type of macromolecule that we're going to talk about are nucleic acids, and they're named as such because you can find them in the nuclei of cells. They are made of puzzle pieces called nucleotides. Each of those nucleotides, or nucleotides, has three parts: a sugar followed by a phosphate group, and those alternate making the side of this double helix or this ladder. And our DNA has that double ladder, whereas RNA, another type of nucleic acid, is a single strand. It still has nucleotides, it just doesn't have them in pairs across from each other. There's also a nitrogen-containing base to round out the 3 components of nucleotides. There are 5 different types. You might have heard this before: A, T, C, G, and in RNA, U or uracil replaces G or guanine. And here is our basic shape of a nucleotide, and I want to point out something else for you here. So our basic nucleotide shape, let me get this right, this is kind of tricky, is right up here. So you've got your phosphate group and your sugar, and the sugar in DNA is deoxyribose sugar, hence the name deoxyribonucleic acid. And then up top in those kind of pink colors, you see one of the bases in DNA. And over here, you see the adenine, guanine, cytosine, thymine, uracil. Those are each of the bases in DNA and RNA, except that, remember, uracil way over on the end there replaces thymine in RNA. So that's molecularly the carbon and the nitrogen and the oxygens and hydrogens that make up each of those nitrogenous bases. Here's a great diagram that shows us kind of the visual structural differences between RNA on the left and DNA on the right. So DNA's got this nice double helix, and RNA is very similar. It's a nucleic acid. It can be found in the nucleus, but it also is going to be able to travel outside of the nucleus. And that's super important for later on when we'll learn about protein synthesis and how RNA has a very vital role in carrying a super important message out to ribosomes for that purpose of making proteins. But RNA is this single-stranded nucleic acid, and instead of deoxyribose sugar, it contains ribose sugar. Carbohydrates are the third macromolecule we'll talk about. They are a great energy source, quick energy source. Structurally, they are made of CHO, C-H-O, carbon, hydrogen, oxygen in a 1 to 2 to 1 ratio. So we've got simple carbohydrates, which are things made up of 1 or 2 puzzle pieces or monomers, and then complex ones, which are longer, and they actually take more energy and a longer amount of time for our body to digest, which you're actually going to see in one of your upcoming labs this semester. So be on the lookout for that. So a great example of a monosaccharide, only a 1 monomer subunit, is a sugar called glucose, C6H12O6. So there's our ratio of carbon to hydrogen to oxygen. And so this is what we call an organic molecule. And the glucose, C6H12O6, is a super important molecule that we're going to see a lot of later on in our chapters on cellular metabolism, specifically photosynthesis and cellular respiration. Some great examples of disaccharides, which have 1 or 2, consist of 2 or more monos, are sucrose and fructose. So those are a little bit longer carbohydrates and take more energy and time for our body to digest. So think about all the glucose, all the sugar, all the carbohydrates that we take into our bodies, and recall how in the beginning of the lecture I indicated that we are mostly carbon, and the structure of glucose right here actually validates that for us. So there's a nice carbon skeleton right here with some hydrogens and oxygens stemming off of it, and so that C, H, and O are really the molecules that make up us in a high percentage. Some examples of some more complex carbohydrates are things like starch and glycogen. So we can actually store energy in these polysaccharide chains, which are based off this monomer of glucose. Some of these polysaccharides actually don't store energy, they are more structural. So things like cellulose that you can find in plants, and then chitin, which is in the exoskeletons of invertebrates, are some nice structural carbohydrates that those organisms produce. Carbohydrates aren't used just for energy and storage, they can also be used for transport. And here's some nice examples of where each of these activities occurs and who can do it and what those molecules look like when you draw them out in their geometric shapes. Our last macromolecule of interest are the lipids.